So I'm going to start off with thanking everybody for joining us today, as we are incredibly excited to have you here. Today's webinar is titled Advancing Non-Viral Cell Engineering to Produce Next-Generation Medicine and How SciNote Keeps MaxCyte in the Forefront of Innovation. We're going to be diving into MaxCyte's approach to accelerating innovations in cell therapy, exploring how their cutting-edge technology is shaping the future. And we're going to also talk about how SciNote supports MaxCyte's R&D efforts, making scientific innovation more streamlined, efficient, and future-proof. And at the end, we're going to open the floor for an interactive Q&A. Before we'd like to dive in, I would like to briefly introduce myself. My name is Kelly, and I am a Customer Success Manager here at SciNote. In my role, I support our partners throughout every stage of their journey with us, from onboarding to implementation to even the long-term adoption and continuing optimization. So that means that I'm here to ensure that your team gets the most out of SciNote, answering your questions, sharing best practices, hosting training sessions, providing ongoing support, things of that nature. I have the pleasure of working with SciNote's team since they began using SciNote. I had the pleasure of working with MaxCyte's team since they began using SciNote. So I know firsthand how amazing their work is and how seamless they've integrated SciNote into their R&D process. At SciNote, we believe that science is more than just data. It's organized knowledge, and that knowledge must be preserved, shared, and built upon. Our mission is to help humanity benefit as much as possible from science, not just today, but for future generations as well. That's why we're proud to provide a digital foundation that supports cutting-edge research across the globe. We're proud to serve a global scientific community, from leading academic institutes to global labs to innovative biotech and pharma teams, and everyone in between. And today, we're especially proud to host a session in collaboration with MaxCyte. So without further ado, I'd like to introduce Dr. Jim Brady, Science Vice President of Technical Applications and Customer Support, and Dr. Ashley Strickland-Dietz, Immunology Scientist. I'm going to hand things over now to you, Jim, to get us started with taking us behind the scenes at MaxCyte.
Great. Well, thank you, Kelly. If you just flip to the next slide. As Kelly said, I'm the SVP of Technical Applications and Customer Support at MaxCyte. MaxCyte is headquartered in Rockville, Maryland, in the United States. And our mission is to enable non-viral cellular engineering through the use of our proprietary flow electroporation technology. Now, before I talk about MaxCyte and how SciNote enables our research programs, I want to give you just a little bit of background on our technology, what it does, and how it's being applied for cell engineering. So if you go to the next slide. Now, cell engineering is a term that encompasses a broad range of applications and methodologies, but almost all cellular engineering approaches are based on delivering molecular cargo into cells. And molecular delivery can be done to address basic research questions, as well as for drug candidate screening. It could be used for bioproduction or cell therapy enablement. And while MaxCyte's technology is used in all these areas, if you just click it one more time, the technology really was developed, or was originally developed, for cell therapy. And this is really the main application area for our end users. If you can go to the next slide. Just to give you a little bit of background on cell therapy. Now, I think everyone's familiar with cell therapy to some degree, and it's shown tremendous promise for treating previously intractable conditions, including a lot of different cancers, as well as metabolic disorders, autoimmune diseases. Cell therapy modalities can be classified into two broad categories. First, you have in vivo cell therapy, as shown on the left. This involves modifying cells without removing them from the body. So typically, this is done by infusing the patient with either an engineered virus or some type of nanoparticle that serves as a delivery vehicle for genes or other molecules. And then on the right, ex vivo cell therapy. In that case, cells are removed from the patient, then they're engineered using a virus or other methods, including electroporation, which is where MaxCyte comes in. And then those cells are infused back into the patient. And frequently there's an intermediate cryopreservation step in the process. And these molecules that are introduced ex vivo, they could include genes that encode tumor targeting receptors, such as CARs, or they could be gene editing reagents, such as CRISPR-related molecules. If you go to the next slide. Now, until recently, most ex vivo cell therapies relied on viral transduction for molecular delivery, and viruses can be engineered as delivery vehicles by removing the viral genes that are required for replication. Then you can insert genes that encode molecules of therapeutic relevance. And then, engineered viruses have a number of advantages. They're very effective for getting molecules in a wide range of cell types, and they often provide long-term transgene expression, which is desirable from a therapeutic standpoint. But there are also a number of disadvantages to using viral vectors. They can present safety concerns. They can be immunogenic, or you can't always control where they insert into the genome. That can cause problems. And there are also significant costs as well as logistical challenges related to the use of viral vectors for cell therapy manufacturing. So if you can go to the next slide. The alternative to viral transduction for molecular delivery is transfection. So transfection actually refers to a variety of methods for getting molecules across the cell's protective outer membrane. And these approaches can include chemical or carrier-based methods, such as lipid-based reagents or charged polymers, but they also include physical approaches, such as mechanical force or microneedles or electroporation. If you can go to the next slide. Now, electroporation essentially involves placing cells and the molecular cargo between two electrodes, a positive and negative electrode, and then you expose the cells to a series of brief electrical pulses. This causes temporary disruptions to the cell membrane that allows the cargo to enter into the cell. And there are a lot of advantages to electroporation. Reproducibility is compatible with a wide range of cells and loading agents. And electroporation has actually been used for many years in research labs. I'm sure many of the viewers have used it at some point in their research. MaxCyte actually took a unique approach to electroporation by focusing on its use for clinical applications. So if you go to the next slide, MaxCyte was actually founded a little over 25 years ago. And this slide highlights some of the significant milestones in our company's history. I won't go through them all, but I do want to point out just earlier this year, we acquired another company, Strickland-Dietz. So that was a very exciting moment in our company's growth. If you go to the next slide, this shows our expert line of instruments and some of the application areas where the technology is used. We have instrument placements in most major pharmaceutical companies. Our customers also include many biotechnology companies, leading academic labs, CDMOs, and CROs across the globe. If you go to the next slide. This figure shows how one of our instruments might be integrated into a typical cell therapy clinical manufacturing workflow. It also highlights some of our unique GMP enabling features and capabilities, including support by an FDA master file, our clinically compliant buffer and consumables. We also provide optimized preset electroporation parameters for a wide range of primary cells and cell lines. Seamless scalability, I'll talk about a little bit later. And also we provide top-level technical support to all of our end users. So if you go to the next slide, so as I just touched on, you can go to the next slide. Yeah, a key innovation of MaxCyte's platform is the use of flow electroporation to scale up the transfection process. Actually, you can go back one slide, sorry. So we can electroporate anywhere from thousands up to billions of cells at one time without the need to re-optimize. And we can maintain the same levels of efficiency and viability across this whole spectrum of scale. So now if you go to the next slide, this just shows that our technology is compatible with pretty much any type of molecular cargo. This could include RNA, large plasmids, protein, as well as small nucleic acids. If you go to the next slide. Here I've highlighted or shown some of the many different molecules that can be expressed following gene delivery with MaxCyte. So you can see this includes targets for drug screening, such as GPCRs or ion channels. You can express recombinant proteins, such as antibodies. You can make vaccines. It's used to manufacture viral vectors. And then for cell therapy, customers are expressing tumor targeting receptors such as CARs and T-cell receptors, and then you can express a variety of gene editing modalities, CRISPR nucleases, base editors, prime editors, etc. If you go to the next slide. So if you look at some examples of the cell therapy applications that are enabled by MaxCyte electroporation, these include, as I just mentioned, CRISPR-based engineering of immune cells and stem cells. It's commonly used to express receptors and T cells or NK cells and macrophages. And as this figure summarizes, the platform has been used to engineer patient cells in a wide variety of clinical trials for treating a variety of diseases. It's involved in both autologous and allogeneic therapies. And at the center of the slide, I just want to highlight that MaxCyte electroporation actually plays a critical role in the manufacturing process for Casgevy, which is the first approved engineered cell therapy for treating sickle cell disease and transfusion-dependent beta thalassemia. And then outside of cell therapy, our technology is commonly used for producing recombinant proteins, or as I mentioned earlier, generating cell-based assays for drug candidate screening. If you can go to the next slide. To support our customers and partners who work in all these diverse application areas, we've built up a team of application scientists with expertise in immunology, gene editing, bioproduction, and other disciplines. And we actually employ more than 20 field application scientists around the world. And they're the ones who work directly with our customers, helping them to obtain maximum productivity or therapeutic efficacy from their engineered cells. If you can go to the next slide. In Maryland, at our headquarters, we have more than 4,000 square feet of wet lab space. And I guess if you click it to bring up the pictures, yep. And we have about a dozen biological scientists in our internal labs, and they are generating data and protocols that we pass along to our field scientists and then ultimately to our end users. And our scientists also produce a lot of data to support our marketing or commercial or product development, product management teams. If you can go to the next slide. This just shows that we do a lot of cell culture work at MaxCyte. And then if you go to the next slide, it also highlights that we have a fully developed, fully equipped process development lab, where we have projects ranging from small scale optimization and multi-parallel 250 ml bioreactors up to we can do full scale production runs and 50-liter large scale bioreactors. Now, because MaxCyte scientists work with so many different cell types and applications, we really need a data management system that's flexible, scalable, as well as being user-friendly. Early on, MaxCyte relied on paper lab notebooks, but as our company grew, we found that data archiving and retrieval became major challenges. And also, when you had multiple scientists collaborating on a project, it was difficult to collaborate effectively when every scientist had a separate lab notebook. So when we eventually decided to make that transition to electronic lab notebooks, well, we initially signed a contract with a large, well-known ELN vendor, and that platform had a lot of potentially useful features, but we soon found a lot of those capabilities really weren't relevant for our specific needs. And we also found that particular ELN had kind of a unusual or unique data storage format that was somewhat cumbersome to implement and use. And also, while that vendor was really good about initial training and onboarding, later they were much less responsive when we had to train new users or when we had other technical support needs. So therefore, a couple of years ago, we revisited our lab notebook needs and we decided to transition to SciNote. So we found SciNote is much simpler to use than our previous ELN, but it still provides all the features and capabilities that our team needs for recording and sharing and archiving data. SciNote also addresses inventory management needs at MaxCyte. We've appreciated the SciNote team has been very responsive to our training and technical support requirements. They also worked with our IT department to port data from our previous ELN, and they're also continually soliciting our feedback and adding new features to improve our user experience. So at this point, I want to hand the presentation over to one of our scientists, Ashley Strickland-Dietz, and Ashley can actually give you a firsthand account of how SciNote enables our internal research at MaxCyte.
Thank you, Jim, and thank you, Kelly. Do we want to go ahead and switch so I can control the screen? Perfect.
Yeah, of course.
Thank you very much. Can you guys see this presentation?
It's loading, just give it a moment.
Okay.
Yep, we can see it now.
Okay, great. So as Jim said, my name is Ashley Strickland-Dietz. I'm the immunology scientist here at MaxCyte. I've been with the company for about 2.5 years now, and I primarily help to develop workflows for engineering various immune cells using MaxCyte's electroporation platform. This morning, I'm going to walk you through how our team uses SciNote to support this work that we do. But first, I'm just gonna explain to you why we went with SciNote. A lot of these were highlighted by Jim just now. Is this working?
It's a blank PowerPoint at this time.
I think. Okay. Sorry, guys. This was actually, there we go. How about now?
Perfect.
Okay, great. Sorry about that. So yes, as Jim said, initially our team had issues with traditional paper lab notebooks. Mainly, it was hard to retrieve data. It was also difficult to collaborate on projects, not to mention they were very cumbersome. And as you can imagine, after 25 years of being in business, there were a lot of notebooks and took up a lot of space. So we made the switch to an ELN, and while our previous provider did make lab notebook maintenance much easier, we found that it had a lot of features we didn't need. And there were also some issues with data storage, searchability, and we struggled to get support and training for new users. We evaluated a number of ELNs, and as Jim said, we decided that SciNote best fit our needs. SciNote is very simple to use. It enables rapid data entry as well as data management. It helps with project and inventory management as well. It's very searchable, and the team at SciNote has also continued to provide ongoing support and training as needed. And also as part of our transition, SciNote even helps to transfer the data from our old ELN to our current ELN. All of which has improved our productivity and allowed us to better support our partners and customers, as well as our marketing, commercial, and product development teams. But in order to support all of our teams, you can imagine that the technical applications team works on multiple projects simultaneously. And so something we also needed from an ELN was assistance with managing all of these projects. And SciNote did have several features that we found to be beneficial for this. First of which is the personalized dashboard that acts as a landing page each time we log into SciNote. Here we can see a list of our ongoing projects along with their current statuses and their associated due dates. And on the right, these due dates are further highlighted throughout the month, both of which is useful for prioritizing tasks based on upcoming deadlines. When it comes to organizing multiple projects, SciNote has a multi-tiered organizational structure consisting of folders and subfolders that allows us to organize our work first by broader application, followed by large projects, then experiments, and then tasks, which makes organizing these multiple large projects much more manageable, especially compared to your traditional lab notebook. And it also lab notebook. And because we can also grant team members access to our different projects, it also makes collaborating both within our team and across teams much easier, since contributors can view and add tasks directly into SciNote as necessary. For actual notebook entries, it's very simple to create a new task. We simply click the Add New Task button and then fill in the provided template with all relevant information. So this can include details about start dates and due dates, notes on the objectives, assigned items, as well as protocols. Another convenient feature that SciNote provides is that once we've filled in this information, if we ever need to perform a similar experiment, rather than having to enter everything in again, we can simply hit duplicate, and this project will be duplicated, and we can just make any necessary changes for the new experiment as well. Once we've completed our experiment, we can move one tab over within the task and add our results. And our results typically consist of fax data, cell counter data, microscopy images, titers, and dPCR data, just to name a few. And because these are already in an electronic format, we're able to simply upload them directly into SciNote, both the raw and analyzed versions. without having to reference a different location. We also don't need to print anything off and cut and paste it as we would in a traditional lab notebook. As you can imagine, this is extremely convenient. It also helps to ensure that our data are all in a single accessible location. And this is something that's extremely important for data management, as well as for any sort of regulatory or auditing purposes. There we go. Once we've completed an entry, we can then move it into review, where we can easily request our colleagues or our supervisor to look over our results as well as to sign off. And this further helps us to maintain proper documentation practices, all while requiring less time and effort than a traditional lab notebook would. And so in addition to managing all of our projects as well as all this data, we also needed a searchable ELN, and SciNote had multiple options for addressing this need. The first of which is through the use of assigned items. So in each task, we always assign items for 6 important variables, including cell type used in each experiment. And when supporting customers, if there is a customer who is having issues engineering activated T cells, we can find this cell type in our inventory and then either click the 100 items that it has been assigned to, or click on the cell type itself and see a list of all of the experiments that have used this cell type. We're able to then go through the relevant experiments and then identify any solutions that might help address our customers' needs. And likewise, we can use this search method for other assigned items, including application type as well as cargo type, just to name a few. SciNote's search features have also proven useful for training purposes. So if we have a team member that needs to learn a new protocol, such as how to culture T cells or activate T cells, they can simply go into the protocol section within SciNote, click on the protocol itself. They can then review the protocol at their own leisure. They also have the ability to view all the experiments that have cited this protocol, which allows them to familiarize themselves with the work that has been done, and also best plan how to proceed with a certain project. SciNote also has a search bar along the top of the screen that enables a more global search. We can search for specific cell types, for example, like T cells, and all entries containing T cell will be shown. If we are looking for data or protocols more specifically, we can filter our results. And we can also use multiple or combine multiple search terms to further refine these results as well. And these search features not only enable us to support customers, but it also allows us to support our content management and marketing teams, with whom we work closely to put out data for use in application notes, as well as webinars and poster presentations. In addition to all of this, SciNote has also helped improve inventory management within the lab. We have inventories for such things as cells, freezers, plasmid stocks, and chemicals. Within each inventory, we can record items along with useful information such as freeze dates, cell type, lot number, concentration, and most importantly, the location, the physical location within our lab. And this is particularly useful for our large storage containers, as well as for items that we don't use very frequently. Another useful feature is the ability to track reagent consumption. So as we use an item from the inventory, the stock amount will be reduced accordingly, which allows us to monitor how much of each item we have left. We can also set low stock reminders that help inform us when we need to replenish an important reagent. And this is important, especially for any shared inventory, and also helps ensure that lab activities stay on track. Additionally, since we support our field teams by shipping experimental reagents to customer sites for demonstrations, the ability to manage our inventory in this way also helps guarantee that customers receive their shipments on time. Overall, since transitioning to SciNote, our team has significantly improved how we manage our projects as well as our data, allowing us to better support our customers as well as our colleagues. And while SciNote has already done all of this and more for us, they continue to provide us with support and also regularly solicit our feedback in order to continue to make improvements, as well as add useful features to their platform. And we are looking forward to seeing what else they'll roll out next. And with that, I'll go ahead and hand it back over to Kelli. Thank you everyone for your time, and I look forward to answering your questions.
Awesome. Thank you, Ashley. Give me 1 second here so I can go back to sharing my screen. All right. Can you see my screen?
Yes.
Awesome. Okay. So yeah, thank you guys so much for sharing more about what MaxCyte is doing for the next generation of medicine. At this point, I'd like to kind of shift into our discussion portion of today's session, which is kind of a good opportunity that we can dive deeper into some of the topics we already covered, and also discuss some new topics that weren't discussed at this point. And I know you guys have already mentioned what led you to decide to go, or what led you to move into going digital with your lab. But I'd like to kind of ask if would you have found or did find that going digital was very successful for your team in addressing all of the challenges that you originally had with being a paper notebook?
Yeah, well, I can start, and then Ashley, I guess you could add on to this. But I mean, I think there's definitely a learning curve. I think people were used to paper notebooks, and it's just natural to, when you're in the lab, to scribble something onto a piece of paper. So entering things with a keyboard in the labs was a little bit awkward for the team. So I think to make that transition a little bit easier, we started using electronic tablets. We use Remarkable, but there are other types of tablets out there. So there you're still writing by hand, but then it creates a PDF that you can seamlessly upload. So that was one of the things we found that made the transition easier. And then I think once we started, saw how easy it was to create templates and share data back and forth, that also helped to get people on board. But I know, Ashley, you can throw in your 2 cents as well.
Yeah, I agree. I think there's always a learning curve, and a lot of us were a little apprehensive making the transition since we were so accustomed to maintaining those traditional paper notebooks. But I think once, practice makes perfect, and once you adjust, it really has made things much easier. It saves on a lot of time and effort. We're not having to write things. Personally, I would always get like hand cramps, and I would get the marks on my hand from all the writing, and this really has eliminated that. And because it's so easy with like tablets and also computers in the lab to just enter data and notes in real time, I'm able to like make a lot more observations, which I think has really helped improve the results and the data that we're generating as well.
I like that you said about you were able to kind of focus more on the observations. I'm probably the first person who's actually brought that to my attention. However, I'm sure that other users are experiencing that. They're just not maybe aware that taking away the paper notebook is allowing you to kind of be more present in the lab. All right. So my next question, and I know you kind of touched on this, Ashley, already, but with your experience with SciNote so far, can you just briefly touch on a product experience or a support experience that has made you, a true SciNote advocate?
Yeah, so I think as we've sort of been mentioning throughout both of our talks here is that searchability is really important. Because we support customers that work on so many different applications and cell types, sometimes our projects can shift focus quickly. So we're required to learn new techniques pretty quickly. Also, pick up projects that might have been worked on in the past that were shelved for a little bit. And so just having the ability to search through everything associated with a certain cell type or a certain technique really has made making those pivots much easier, much faster. We actually have an experience where a colleague of mine needed to pick up a project that was done many years ago before we had transitioned to an ELN. And it actually became a team effort, sort of wading through all these years worth of notebooks and trying to read the handwriting to sort of identify the information we need. So I think that feature has been really nice.
I hear that time and time again. Actually, I've experienced that in my family as well, where you have to go back to reading a paper notebook, and handwriting is something you have to deal with. How they've recorded their notes, because it's not usually standardized in a notebook where you have that template that you can follow in SciNote. All of that kind of just makes it 10 times harder to kind of figure out what they did and how they did it. All right. And I also like to I'm going to go back on a feature or something that Ashley said regarding that you like to upload all of your files with your work. And a feature that I just want to kind of briefly give its time to shine here is something that could help a lot with those who are adding their files with their work. It's one of my favorite features right now, and it helps make file management 10 times easier. And that is our newish feature, the SciNote edit. So as SciNote edit is something that you would download onto your computer. It does work in the background, but allows you to work with all of your files that are directly inside SciNote. So then you can effortlessly open them in their native software applicants, make those edits, and then save those changes seamlessly back into SciNote. So you're seamlessly keeping your files with your workflow, and everything is in one place. So I just kind of wanted to also highlight that, because that is something that MaxCyte is doing with keeping their files with their work. One way to just have a leg up there is also using our SciNote edit along with that. And kind of to transition into that, are there any other softwares or tools that you use in your day-to-day with your ELN? And how does your team streamline those interactions between the tools and SciNote?
Yeah, I mean, well, I can touch on a few, and Ashley, if you have others, feel free to join in. We talked about the use of the Remarkable tablets, but we have FlowJo software for analyzing flow cytometry data, and we have molecular biology tools for analyzing DNA sequences. We have printers. We have statistical software we use. So, as you said, Kelly, that SciNote edit feature makes it easy to transition back and forth between these other software platforms. And our IT person wanted to highlight too that the use of open APIs also basically provides limitless capabilities. So any task that you set up in SciNote can set up a API trigger out to another system. So this seamless integration we found to be very powerful.
Yes, and I pretty much agree with Jim. I know some of us we're still trying to adjust. We're getting used to SciNote edit. And as Jim said, with the API, there are some softwares and certain instruments that we use in the lab that I think we're working with SciNote to sort of have that seamless integration from one instrument to the other. And so I'm really looking forward to that as well.
Yes, because we know that, you don't just use SciNote, you're going to be using SciNote and other things. So we love to hear about what other labs are using with SciNote, and that's where also SciNote edit came to make sure that we make things seamless there. Right. What advice would you give to researchers or biotech teams starting to explore non-viral engineering methods?
Well, I think you have to think about the final picture. What's your clinical process going to look like? And you want a platform, ideally, that's going to take you from basic research through to process development and into the clinic without having to make awkward transitions or without having to re-optimize at each of those transition points. So there are a lot of, basic research methods for transfecting cells, but a lot of those aren't really suitable for clinical use. They can't be scaled. So, one of MaxCyte's key selling points is that we do offer that seamless scalability. We have instruments that can address the needs of basic researchers on up to clinical instruments, but any protocols or methods you develop at the smaller end of the spectrum can be seamlessly translated into the clinic at large scale. And also you want to think about, again, or is the platform you're using or planning to use, is that something that's been validated in the clinic? And what kind of technical support are you going to be getting when you adopt that platform? So those are all questions I would ask people to think hard about before they launch into a non-viral engineering approach, and maybe find out years down the road that it's not necessarily translatable to the clinic.
Sticking with the advice topic, what advice would you give companies considering to switch to a digital solution to manage their data? And are there any best practices that you think that they should follow?
Yeah, and again, this is something Ashley can jump in on, but I think one thing we learned is you want to spend some time up front or initially to map out your data structure, your master data or metadata, whatever you want to call it. You map out your approval workflow ahead of time. And then obviously working with the SciNote team to make that implementation as seamless as possible is really important. But Ashley, again, if you could think of anything else that would make that digital transition easier, please share your
Yeah,
thoughts.
yeah, it really depends on the company's needs, and really they should think about that before they embark on this journey. But also when considering an ELN, I would highly recommend that they make sure that they find a partner that is willing to support them throughout the entire process and provide that ongoing support and training as needed.
Yeah, something, the ELN's not going to make your coffee and take notes right from your brain and put it on the piece of paper. At the end of the day, it is, work that you're going to have to put in, and what you put in is what you get out. But there are a lot of best practices I know that the CS team has compiled. We've actually created a task that is shareable. So if anybody who is interested in seeing those best practices right now, just reach out to us and we'd be happy to share that. I'm going to just share some of the key best practices that we have come across. However, this is not just all of them. We do have a lot more. But with that, one of the big ones is defining a clear internal standard operating procedure. You have them for your work, but I also believe that having them for how you're going to utilize SciNote is very helpful. It allows things to be consistency. It ensures that you're efficient in how you're using it, and ensuring that you're following compliance across your organization as well. With that, establishing a reoccurring quality review and optimization cycle to regularly assess and enhance your use of the ELN. It promotes continuous improvement and long-term success. So don't just think once you start using and SciNote that the way you're using it now is the way you're going to use it down the line. You can always improve that, work with us, and we can help you improve that to make sure that things are moving as smoothly as possible, depending on what stages you're in as well. Another one is, which they mentioned, but is continuously invest in continuously team training, onboarding, and support. That's what we are here for, is to make sure that you have the tools to be successful. So as your team grows, as we get feature updates are being released, just making sure that you are up to date on the knowledge of SciNote can help make sure that you maintain being productive and utilizing the feature to its full capabilities. Collaborating with your customer success manager was another one that kind of goes hand in hand with the one above, but setting measurable success metrics from the start is very helpful, as this helps align your goals and it helps you track the impact of SciNote on your operations to see that what is working and what's not, and what we can do to improve your usage or what you're trying to get out of going digital. And then lastly, this I've seen from a lot of my good big groups, is forming a dedicated SciNote committee with your organization and champion adoption. So you manage feedback, you drive internal initiatives that are related to the platform. You kind of just work together internally, but then also work with the SciNote team to make sure that everything is moving in the direction that you want. These are just some of them, like I mentioned, that are best practices. However, there are a a plethora. We have a list of them that we can definitely share with you. I know that one of a question that did come in was how to motivate and encourage your scientists to use the ELN. I like to briefly just mention that sometimes people don't want to change the habits they already have. So a lot of times you have to come in and say, starting at this date or starting at this project, we're now going to put everything inside SciNote. So having that SOP so that users know what is needed of them, how they're going to be utilizing what is going to be inputted into the ELN, takes that guessing work out, so that way they know exactly what they need to do, and then they know exactly when they need to start using the ELN. No one ever wants to be the bad guy to say that you have to start enforcing it, or you have to make sure that by this date, all of your data is up to date. But at the groups that have those checkpoints and those deadlines tend to be the most successful. All right. And I'm going to move on then to our next question here. And what tangible benefits have you seen since moving away from that manual paper-based system?
Well, I think, again, the key benefit is not having to dig through mountains of paper lab notebooks every time you need to find data. I think collaboration's easier. Data conformity, if that's the right word, is really important and much easier to enforce with ELNs. But again, Ashley, what are your thoughts there?
Yeah, and also data mining is very useful. I think some tangible items. We work very closely with the content management team to put out various data sets, and they've really found it easy and useful to just be able to go into SciNote and search, and then search across the team's work and pick those data that can be used to support our customers.
A lot of times, the benefits that each group sees can be different because it all depends on what your goals are for going digital. But I will briefly just kind of mention on some of the ones that we have seen or other groups have mentioned that they have experienced since going digital, and that is improving data organization, like you mentioned, and having centralization, making it easier to find, manage, share that information with not only your team, but sometimes outside collaborators as well. It enhances collaboration across your team and departments. It increases data integrity and traceability. It allows for time saving and greater workflow efficiency by automating routine tasks and reducing duplication, especially in work that you might do time and time again over and over. It allows for stronger compliance and audit readiness. And it also helps improve data security. I hate to say it, but that paper notebook, if something were to happen, someone could easily swipe it, or worst case scenario, something happens in your lab and now that paper notebook is ruined and you won't be able to retrieve that data because you can't no longer read that notebook or have that notebook. But overall, transitioning to a digital system, I apologize, my dog is now barking in the background. But it helps groups work smarter, stay aligned, and focus more on the science. Like Ashley said, how she was able to now be more observant in the lab allows you to be more present. All right. So if there's nothing else and we don't have any other questions, we're going to kind of wrap things up here now and give you guys some time back in your day. So I want to thank everyone on behalf of SciNote and MaxCyte for thanking you all for joining us today. If you didn't get a chance to answer or ask any questions, or you feel like your questions weren't answered during the session, don't worry. We'll follow up with an email or reach out to our team at premium@scinote.net. Also, a full recording of today's webinar will be sent to you via email, so feel free to rewatch it or share it with your colleagues. We also have MaxCyte's oh my goodness, I forgot the word here, but that is okay, and how to contact them. And then we have QR codes so that way you can follow them on LinkedIn. So again, once again, thank you for your time and your interest, and have a good rest of your day today and a good rest of your Friday and weekend.
Thank you.
Thank you, Kelly. Thank you, everyone.
All right. Bye-bye.